Hi guys, we're going to talk through landscape genetics today, and this lecture gets a little bit tedious at times, but I want to make sure and give a little more background than the book does, just review of some of the elements of genetics and selection processes that are going to influence how we view this from a landscape perspective. So the debate really continues over the relative importance of genetics and demography in landscape scale conservation. So as a backdrop of this, keep in mind that habitat loss or degradation we have covered and understand is really the overriding factor. That human resource consumption and population growth are the roots of most of our landscape challenges. So when we seek to understand how gene flow interacts with landscape structure and the key differences between population genetics and that is the key difference between population genetics and landscape genetics, we know that landscape geneticists may study the impact of things like resistance of the matrix as a key factor in isolating populations, more so than distance alone. And we also recognize that there maybe are not that many ways we can intervene in remediating genetic problems. So we can argue that genetics are more of a diagnostic tool than a remediation tool. But others have argued more strongly that genetics is relatively unimportant because demographic and habitat factors are always the first causes of declines, and that genetic problems are a consequence, not a cause. If we accept that premise that demography is more important than genetics in determining the fate of populations, we might still find that genetic tools are some of our best route to a demographic understanding and a means for evaluating dispersal patterns. We've already talked about the challenge in actually tracking and understanding dispersal when we want to understand source-sink dynamics. So just a couple other notes here, how microevolutionary processes of gene flow, genetic drift, natural selection all interact with a diverse landscape to shape patterns of genetic variation across the landscape. This is a sort of a conceptual definition of what landscape genetics is all about. And landscape geneticists, as we already said, may study the impact of resistance within a matrix, isolation by resistance, as key factors in isolating populations. So what are the differences between genetics versus demography? We know that several studies have shown that cheetahs exhibit very limited genetic variability. Is variability a crucial threat to cheetahs' persistence, or is the lack of variation simply a reflection of the demography within that species? Let's review quickly just a few of the principles of natural selection, which I'm sure you guys have learned many times in many different courses. But it's important to remember that natural selection is based on random chance and random chance alone. No species can will itself to have adaptations that help it better fit its environment. So as it relies on random mutation to serve as the source of genetic variation, it's essentially a lottery system. The odds of adapting to changing environments really relies on the number of mutations and the chance of a random new allele that offers some adaptive advantage in that changing landscape. So it's a numbers game at the end of the day. Population size effectively equals adaptive capacity. So we know that the rate of evolutionary change in a population is going to be proportional to the amount of genetic diversity available. Genetic variation provides the raw material for future adaptation, and loss of diversity will generally decrease an organism's ability to cope with environmental changes. Where does genetic variation come from? It comes from mutations that can express themselves in many different ways. We have gene mutations that are just errors in coding. They can alter only a few base pairs or alter a single protein. And we also have larger mutations that generally result in a non-viable offspring. But chromosomal mutations can be another source of diversity, particularly in plant species. So these affect the number of chromosomes or the number and arrangement of genes on the chromosomes. They tend to be lethal in animals. And just as a review, an allele is a specific version of a gene. One allele can exist for certain genes or hundreds. And if a gene has several major alleles, the population may be heterozygous or homozygous in several different ways. So a gene may have one dominant allele and two recessive alleles. If a population has two or more alleles of a given gene, it's called polymorphic. And we'll get into the different types of diversity we might measure within a population from a genetic perspective. So we can view variation in several different ways. It can scale up neatly in a hierarchical fashion from variation within the individuals, and all variation really, individuals are the source of variation. But we can scale that up to view variation within individuals in a single population. We can look at variation among populations and even among metapopulations. So what does that variation within a population look like? We know that there is neutral genetic variation, genes that are not under adaptive selection, sort of the evolutionary baggage out there, and they change at different rates, but can be a helpful source of information when we're looking at relatedness among organisms. And then we've got adaptive variation for those genes that are currently under selective pressure. And we're most concerned about actually being able to preserve adaptive variation within a species, but often we don't actually know which sections of DNA are adaptive and which are neutral. So gene frequencies in a population, we know that they are likely to change over time, and there's several different ways that they change. Mutation obviously is the source of change in gene frequencies, and when we might see a new allele introduced to a population. Natural selection, we know, is the active adaptive change that we might see in allele frequencies within a population. But then there are also random processes that can be at play that change gene frequencies. And it's these random processes that are more likely to be influenced when we have a small or isolated population. So genetic drift is the change in gene frequencies and heterozygosity due to chance alone. And again, that's something that we're more likely to see in a small population. And then things like unequal family sizes, some mating systems, changes in population size, all of those are random processes that are going to influence gene frequencies and keep them from being normally distributed among the population. And then, of course, gene flow is another way we can see processes that affect our distribution of genes. So gene flow is supported by immigration and emigration between populations. The loss of gene flow between populations can lead to genetic divergence over time. And we know that ultimately that's a mechanism for evolution of new species or new subpopulations. So a few things to remember about small populations are that the chance of inbreeding in small populations between close relatives increases dramatically. It doesn't necessarily change allele frequencies, but we know that the frequency of homozygosity increases dramatically with small populations. And for that reason, the expression of rare alleles and even deleterious genes, genes that are going to decrease fitness and survivorship, have more of a likelihood of being expressed in a small population, and particularly among related individuals. So for that reason, inbreeding often leads to decreased fitness, and we've seen this in captive populations, small populations. We've seen it in the royal family, where spouse selection was limited to other royalties. Genetic drift, a few more important things to remember about genetic drift. We know that the random fluctuation of gene frequency, genetic drift is the random fluctuation of gene frequency over time due to chance alone. It can lead to loss of variation rather quickly, and it is one of the greatest conservation concerns, and I would say one of the preoccupations of landscape geneticists. It's the only mechanism to recover variation lost to genetic drift is mutation, and we know that that's a lottery system in itself. Gene flow from immigration via natural or assisted means is important, and as habitats become more fragmented, gene flow is a way that we can counter the effects of genetic drift. So gene flow is the movement of genes from one population to another, while genetic drift tends to increase the differences among populations. Gene flow actually decreases those differences. It's a way where if gene flow is uninhibited, we would expect to see the same distribution of alleles into connected populations, or very similar. And so it also offers a way for us to assess the level of gene flow, and by looking at the allele differences between two populations. So genetic similarities among populations can be due to more recent common ancestry than gene flow. So genetic measures of gene flow really are sort of cumulative effects of contemporary and past events, and that's one of the challenges in using some of our genetic information to draw inferences about things like the level of gene flow. It's really hard to dial in a timeline for when evolutionary for these genetic markers when things changed. So what is the relevance of all this to landscape ecology? We know the study of landscape genetics integrates topics that we've already discussed. It affects the landscape connectivity on individual movement and dispersal. There are population distribution and dynamics, relationships that can be inferred from genetic information, population spread. We can look at the asymmetrical flow of genes as a way to help identify source sink dynamics within a population. It can help us in understanding how genetic variation is distributed within and among populations, and that's an important factor in species conservation and management. So your book talks a little bit about the F statistic, the assessment of the degree of genetic differentiation among populations. And it is important to sort of understand how to interpret those F statistics. This is a measure of genetic relatedness that's been around for a long time and is important as a tool in landscape genetics. So the lower F statistic means that there's little genetic differentiation among populations, essentially that they are genetically the same. And if we get up to an F statistic of over 0.25, we know that there's a great genetic differentiation between populations. And the measures between those two, you can see the scaling here in table 9.2. So the book gives a nice primer on the tools that we use to measure genetic variation within and among individuals. The genetic markers that it discusses include microsatellites. Microsatellites are short nucleotide sequences. They usually consist of maybe 2 to 6 base pairs that repeat many times. The number of repeats can be variable among individuals, and it can give rise to quite a few alleles. When we're looking at microsatellites, heterozygotes can be distinguished from homozygotes, and that can be helpful for assessing within population variation. We know that the majority of microsatellites we might be looking at are going to be selectively neutral, so coded in what we call junk DNA. And using kind of these junk DNA or non-coding regions can help to isolate the differences in gene flow and genetic drift rather than those that are under selective factors. So they might actually be preferred when we're most interested in looking at genetic drift and gene flow. And we know that those two forces are more likely to be influenced by landscape factors. And they can be useful in tracking dispersal events. They require a lot of lab work. They're labor intensive, so aren't always the preferred marker method. The amplified fragment length polymorphism represents a sort of whole genome approach where tens to hundreds of markers are generated to give individuals more of a DNA fingerprint, a little bit like a barcode. And this is a coarser approach, really looks for presence or absence of gene segments. It doesn't identify heterozygotes for that reason. And then single nucleotide polymorphism is just looking at a single nucleotide substitution at a base. So adenine, thymine, guanine, or cytosine. For another within the DNA sequence. So we can look at these with neutral or adaptive DNA. This approach has been important to landscape genomists that seek to identify landscape factors that are responsible for adaptive genetic variation. And I thought Table 2.1 did a good job of sort of reviewing the advantages and disadvantages of some of these different genetic marker approaches. So let's talk a little bit about genetic variation among populations. We know that variation among distinct populations are a critical component to overall genetic diversity within a species. We often see localized divergence. We can see that from local selection processes with slight differences in the environment, but also due to random processes that we've already talked about. But it gives us a sense of the spatial distribution of variation, and it can help us really define areas of interest for conservation. Some of the metrics that we might look at when we're thinking about variation among populations includes things like allele richness, the number of alleles per locus, allele frequencies, the relative proportion of alleles within a given population, and then measures of heterozygosity. I know you all are familiar with the Hardy-Weinberg equation. This is an important equation that is used in landscape genetics for a population that is assumed to be at equilibrium. Within the Hardy-Weinberg equilibrium equation, we can use the observed frequencies of two alleles and calculate the expected genetic genotype frequencies within our population. And so it gives us a baseline for which we can compare our populations to see if they are indeed at equilibrium or if there are influences at play that are preventing the population from being at equilibrium. A few of the challenges we might face when we're using genetic markers as a tool to understand landscape dynamics is that often the statistical analyses that test the null hypothesis that the populations are non-isolated, and due to low power in a limited number of samples, it can actually be difficult to refute that null hypothesis in cases where even little gene flow occurs. Another issue may be that which genetic markers are being used. We know that neutral markers are less likely to capture the same type of genetic variation. It can be more, it can be less as adaptive markers. And the vast majority of cases, we don't actually know if we're testing neutral or adaptive variation. One of the metrics that's discussed in the book is the effective population size, represented by NE, for a population. This is an idealized concept of a population that typically considers a large, randomly mating population with non-overlapping generations. We know that real populations don't meet all these criteria. Generally, but this again gives us a baseline against which to measure how our real populations are interfacing. So ratios of effective population size can be affected by fluctuations in the size of the population, variance in family size and taxonomic groups, and unequal sex ratios. We consider the effective population size in part to calculate the probability of losing heterozygosity through genetic drift. So how do populations diverge? We know that a population is a group of organisms of the same species that interbreed frequently and share a particular space. The small local population we may refer to as a DEAN, a gradual shift in any genetically controlled trait we might refer to as a cline. And then we know that species are similar in related populations. So what leads to evolution and divergence once populations are isolated? And there's actually an interesting tension in my mind between our preoccupation with making sure that populations are not separated and our knowledge that most species divergence and speciation events were caused when populations were isolated and separated. And often these speciation events occurred with small founder populations. So what we know to be the source of biodiversity, we also are actively concerned with, isolating our populations. So what leads to evolution? In the past, we know that divergence of populations once they're isolated is how many speciation events occur. That generally means that there was a primary isolating barrier. It might be that a small founder population found their way to an island and were separated from the mainland population, a new river, a new mountain range that separated populations. All of those are natural isolating events. And we know that humans have caused many non-natural or anthropogenic isolation events in the landscape. Two isolated populations may become genetically fixed, and mutation may introduce new variation into the population. Small populations, genetic drift, founder effects, all are going to help with that divergence of two populations. And then other forces at play are non-random mating and natural selection. So we've talked a little bit about the statistic to determine the degree of genetic differentiation between populations. We can look at the extent to which the average expected heterozygosity within a population deviates from the expected for the entire metapopulation. So we know this as FST, and this is the F statistic of greatest interest to population geneticists. Conservation of genetic diversity. We know that conservation, guided by an evolutionary perspective, seeks conditions that will allow populations to continually adapt to inevitable changing conditions. So maintenance of diversity within population is meant to meet three objectives. Maintenance of viable populations that can avoid extinction, maintenance of the ability to continue adapting and continue adaptive change. And again, that relates to the numbers game of having enough interbreeding individuals for random genetic mutations to occur. They could be adaptive. And then maintenance for the capacity. So a lot of our discussion has been about assessing populations, but this assumes that we actually understand what constitutes a distinct population. Another way we can use genetic information is it can help us understand what actually constitutes a population. So we can apply various statistical approaches to see what spatial genetic structure occurs within a population and gain an understanding of which individuals are actually interbreeding most frequently. A couple of different analyses that we see are the Bayesian genetic clustering. is each population is hypothesized to be in Hardy-Weinberg equilibrium and to exhibit that kind of linkage at equilibrium. But Bayesian cluster analysis without admixture assumes a few things. It assumes that individual populations diverged from a parent population over time. Barriers to gene flow followed by genetic drift are assumed to have given rise to the genetic structure that could be identified as a result of differences in allelic frequencies among a number of populations. So individuals can be assigned probabilistically to a population based on those genotypes. And this is one way that we might actually try to define populations out of a broader sample. Bayesian cluster analysis with admixture assumes that individual populations were likely derived from some unknown number of parent populations over time, and that those individual genotypes reflect a mixture of alleles that originated from one or more of these populations. Mixture proportions can help us understand relatedness among existing populations or even infer ancestry or origin. We can infer that the sample group now represents at least two distinct populations. That's the dotted line in the middle of the current sample below. And so I've got an example. The book gave a really interesting example of a canid study in Ontario. It was an analysis of interbreeding coyotes, gray wolves, and eastern wolves in a region of Ontario. And what we found was that although gray wolves and coyotes seldom interbreed, they both breed with the eastern wolves. And a Bayesian cluster analysis with admixture confirmed that there were three distinct populations. It reinforced the regional affiliations, coyotes to the south, eastern wolves in the middle, and gray wolves to the north. The analysis also showed that eastern wolves were serving as a genetic bridge, allowing for that kind of hybridization to occur among the three groups. The canid study used also the multivariate ordination technique. These kind of techniques offer an alternative when the assumptions of genetic equilibrium can't be met. There are several different multivariate analyses that can be used, including the one we're looking at here, the principal components analysis. We could also use principal coordinates analysis or non-metric multidimensional scaling. But here, the principal components analysis were used really to cooperate the inferences from the Bayesian clustering analysis in the three canid populations. The first two principal components were able to differentiate individuals associated with the three regional populations. There was a greater degree of overlap between the coyotes and the eastern wolves that reflects that higher degree of hybridization between those two species. So some of the current focus of landscape geneticists are uncovering the landscape correlates of population genetic structure, really understanding those dynamics between genetic structure and the heterogeneity of the landscape. Assessing functional connectivity in the landscape. So using this genetic information as a tool to help us understand how populations are or are not connected and allowing for gene flow. Characterization of source sink population dynamics, And we can look at that through differential movement of alleles among populations, and also determining the relative influence of current versus past landscape structure on genetic structure. And as I said, this can be really difficult to assess because that temporal element is often the trickiest to nail down when we're looking at genetic differences. And then also evaluating the potential for adaptive responses to climate change. So the latter of these two, we're not going to go into detail, but please take the time to read those sections in your book. So some of those landscape correlates of population genetic structure, we know that we can explore the relationship between landscape structure and genetic processes by looking at the degree of correlation between specific landscape features and population genetic structure. So it's easy to imagine features such as elevation and slope and the degree of habitat isolation and how those might positively or negatively influence genetic distance between populations. So rather than relying on empirical data reflecting things like dispersal and immigration and emigration across landscape patches, we know we can use genetic information to relate gene flow patterns to landscape structure and draw inferences about functional connectivity. Landscape geneticists have contributed to our understanding of really a new paradigm in landscape ecology, our understanding of isolation by resistance rather than simply isolation by distance. And a lot of the same modeling techniques that we've already talked about can be used to help us understand that. Landscape genetics has really been a boon in the study of functional connectivity, and especially for species that we rarely observe dispersal or migration events, or where it's really hard to measure these types of events. Insects and plant pollen, seed dispersal, pelagic larvae, others are really practically impossible to be able to mark and trap to understand immigration and emigration, gene flow, movement of individuals and genes among populations. So genetic markers have allowed us to actually understand this much better. So the ecological and genetic consequences of some of these barriers that we might see are one of the preoccupations of landscape geneticists. This figure actually shows the link between some structural changes and the ecological consequences that we might see, which leads to genetic consequences. We know that road-related habitat loss may reduce the effective population size, which, and while the barrier itself caused by roads can also reduce gene flow, leading to reduced genetic diversity. So while direct road mortality leads to both of these genetic consequences. When we look at the ways we might assess barriers to gene flow, it's really important to remember that there's often that generational time lag involved. Sometimes over 100 generations before we actually see that reflected in genetic markers. So detecting these barriers in real time can be actually really difficult. And then the converse can be true as well. The impact of past barriers that have been removed can persist for long periods of time. And this is, of course, good news for conservation that we often have some time after these disturbances in the environment before we see a genetic reaction to those. So the relative effects of habitat amount versus fragmentation, there was a really interesting study done on red-cockaded woodpeckers. We often refer to these as RCWs. This is a modeled landscape study that looked at the effects of fragmentation on potential genetic structure and a fixed habitat amount. So we looked at a fixed amount of mature pine forest. It was fixed at about 7% of the landscape, and then lower quality secondary growth forests varied here from 10% to 70%. What we know about this particular species is that they're a habitat specialist that requires mature longleaf pine forests. They need the older trees, older than 80 years old, that have a bit of heart rot in order to excavate their cavities. Rather than dispersing, young birds return as helpers and help to raise the next generation in the following year. And additionally, the species are known to be very poor dispersers. They're unwilling to move across gaps over about 600 meters. And they and more than a handful of kilometers to move into suitable habitat. So there's a high potential for inbreeding depression amongst these populations. In this case, the researchers analyzed the hypothesized effect of population size and the degree of genetic variation based on the various habitat configurations. And I really liked this quote that the authors of this study offered, and I won't read it to you, but they do come away with some really interesting takeaways from that study. Source sink population dynamics are something that we can use this genetic information to track, and there's some really interesting studies that have been done to this effect as well. So Bayesian genetic structure tests were used to identify source and sink populations of mountain lions in the Sierra Nevada mountain range. There were found to be 5 genetically distinct populations. In this figure, the arrows reflect the direction and the rate, the thickness of those areas of recent migrations. They show that there is asymmetry in the flow. Sources here are net exporters, while sinks are net importers. And what we're seeing in the future of landscapes genetics is really a convergence of some of the various approaches to how we study adaptive genetic variation. So landscape genomics really holds a lot of promise for being able to evaluate the adaptive potential of species in light of climate change. But all of these different disciplines are offering us some insight into how to manage populations over time.